Good evening. Welcome back to another episode of Human Physiology Hybrid. All right, so on the screen in front of you have transport across the plasma membrane. So this chapter, well, particularly this section of the chapter, focuses on the transport across cell membranes as a mechanism for homeostasis. So in chapter 1, which I know in this particular semester I did not cover, face-to-face I do, we go through the themes, and feedback loops are a big, primary actually, mechanism for homeostasis. This is another one at the cellular level. Transport across the cell membrane. For example, cells have to regulate, right? The concentration of nutrients and the waste. How do they do that? What structurally is involved in knowing? How does a cell know what nutrients to bring in? And how do they do that? Each of those nutrients and molecules have properties of their own, such as polarity, hydrophobicity, or philic or lipophilic. So there's a variety of vitamins and minerals and macromolecules, subunits, monomers, that have their own properties, and the cell needs a lot of those. What is it about the cell that it's able to take in and recognize each of those things in the right amounts? Because it won't take in more than it needs. Generally, there's not enough space in there to store everything indefinitely. That's what liver and adipose tissue and other things are for, storage of certain proteins and sugars. So it's worth thinking about. Likewise, waste, right? Some things need to get out of the cell. Once we've used those nutrients, or converted them for use, what does the cell have in it, or about its membrane structure anyway, that it can get rid of them? And likewise, you might think of this when you think of medicine, Tylenol, cancer medication, anything. It's all about marketing it to the cell. So how do you, when you take that pill, how do you get that active ingredient in the medication or the antibiotic to get to the cells that it needs to, and so that those cells that need it will actually take it up, and so it can have its healing effect? It's the craziest thing. I was a kid. It was magic to me. Someone had a pain in their ankle because they sprained it playing baseball. Here, take a couple Tylenol. It's like, what? Someone had a headache. Here, take a couple Tylenol. Here, fell on my knee. How does Tylenol know to go to the knee or the head or the ankle? It was just crazy to me. Well, now I know it enters circulation and it goes everywhere. But you're only going to feel the effect, of course, where the pain was. It relieves pain, and like ibuprofen, decreases inflammation all over. But you're going to feel the relief wherever it was originally inflamed, right? Now it's an anti-inflammatory, so now it's uninflamed. So this is kind of the study of medicine there. Also biomedical sciences, pharmaceutical sciences, and research. It's all about how to get the right kind of medication to the cells in a way that the cells will take it up. And of course, we do that on a pretty regular basis, obviously. So maintaining concentration levels as well. That's another big topic for homeostasis. There are some things that the cell doesn't want to lose, needs to keep, and some things that it never wants to take in, it always wants to lose. So that takes sometimes an active form of transport, a little more energy than just allowing things to come in and out that it already wants and that are able to. Some things need a little help, and that takes a form called active transport we'll be looking at as well. A good example is the sodium-potassium pump, which we'll talk about in this lecture. It's a carrier pump, and it regulates the amount of sodium. You're told on nutrition labels anyway to watch your sodium levels, and it's true. We tend to get too much because it influences high blood pressure. Oh, that's going to be a good question for discussion sometime. So what is it about sodium that causes high blood pressure? We can kind of apply some of what we're going to learn today to that phenomena. But anyway, you do need a lot of sodium for proper nervous system and cell concentrations of water. So sodium is very important, as well as potassium. So there are pumps and carriers just for that reason and process, which by now I'm thinking you've gone through the slides and or read the chapter, and so, or at least those sections, and you know a little about. All right, enough said. Let's get on with this. We're already at 5 minutes of introductory remarks. Sorry. So phospholipid bilayer is selectively permeable, simply meaning that not everything can cross it, even though it needs to, and not everything can cross it, even though we don't need it to, or wants to and we don't need it to. So you can just read through these slides. I want this to be fast, and you got some definitions there. The main thing here is active and passive. And of course, if it's active, it requires energy. Any active lifestyle requires energy. Any passive, sedentary lifestyle requires no energy. Well, cell transport somewhat relates to that, right? So passive, these are shapes of molecules, sizes of molecules are such that they easily and on their own can pass right through the cell membrane. And then active transport simply means that they don't pass through naturally or easily down the concentration gradient, so they need a pump. And that word pump ensures, just like a water pump, you're going to need energy to expend that. So which mechanism is used depends on the type of substance it is that needs to get in or out. And so we do have a permeability. It's not just a solid surface that protects the cell from the outside elements, but it is a cell that is dynamic and living, so some things need to get in, some things don't. So the concentration of a substance within the cytosol and the extracellular fluid. These two are dynamic and ever-changing concentration, and the cell has to maintain a certain concentration regardless of what that extracellular fluid is. So the passive transport process, in short, is simply the movement from an area of high concentration to low concentration. You've seen this if you've seen Kool-Aid, if you've put food color in water, if you've sprayed perfume in a room. It doesn't take long for it to fill the entire room, or someone gets out their lotion, and the odor, the aroma diffuses across the air medium, or in this case, the aquatic or water medium. This is a law of physics. This is Brownian motion. It's random Brownian motion. Things just bump into each other until there's, as long as there's room to bump and move, they'll be set on a trajectory. Once hit, boom, and it'll just go in one direction until it's acted upon by another force. Boom, and then it's acted upon by another force. Boom, 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 boom. So you have a lot of interactions in here, and they just naturally go from an area of high to low concentration. That's passive transport. We call that kind of transport diffusion. So the movement of those solutes from high concentration to low. So right here in this first picture, it's a very organized system. It's organized because it took effort. We put this plastic divider, and you have a high concentration of sugar here, and you have a low concentration of sugar here, which means you have a high concentration of water here and a low concentration of water here. Passively, solute particles or water all want to move down their concentration gradient. So sugar wants to move in a direction as it bumps into itself down its own concentration gradient. Likewise, water wants to move down its concentration gradient from high concentration of water to low concentration of water, and from high concentration of sugar to low concentration of sugar. So we move it out, and there it goes. Quantum mechanics, right? No two things can take up the same place at the same time. So sugar is now moved over here, water is moved over here, and we're going to reach this equilibrium, a very low energy state. This is not typically the way your cells or your body function. We need to have certain things in higher concentrations. And as I said in the very beginning, this is organized, and therefore it's going to require energy. Now, the movement is passive, but keeping a living system this way takes energy. So passive transport process, simple diffusion. These are typically done by molecules and able to be done by molecules that are nonpolar. Why? Right, if they're nonpolar, they're hydrophobic. And what is the plasma membrane made of? A phospholipid bilayer, which is hydrophobic. So like dissolves like. So nonpolar molecules, steroids, steroid hormones, these things, cholesterol, they're recognized by the cell very easily, or whether or not they're recognized by the cell, they're able to diffuse in right across the membrane from high concentration to low concentration, from high concentration to low concentration. So simple diffusion of a nonpolar molecule can go through phospholipid membrane. It must be nonpolar or very, very small. So there are some very small ions. Sometimes water leaks across. Some small ions leak across, but that's just it. They leak. They don't really come in mass like this. They just kind of boom, 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 boom. They bounce around those phospholipid tails. So other nonpolar solutes, oxygen, carbon dioxide. So you want to think in context of your lungs, right? And the capillaries that surround your lungs and the movement of oxygen and CO2 across your lungs. That's a form of passive transport, and it needs to be passive, right? Lipids, as we just said, steroids, hydrocarbons, fats, pass straight through that lipid bilayer. All right, so facilitated diffusion is still a form of diffusion, even though it requires a protein carrier or some help. These are typically channels, kind of shaped like donuts. All they are just large holes for those larger molecules or for ions to pass through. The reason this is somewhat of a tight membrane. Very small things might bounce around through there, but we need things in mass sometimes. We need larger quantities in bulk to travel through to replenish the cell, especially when you're in a state of, say, dehydration and you need a lot of ions quickly. So facilitated diffusion involves charged or polar solutes such as ions, glucose, which even though it's a simple monomer, it's still too large to just come right across the membrane. So they need a little help through a carrier or a channel. So the reason it's still diffusion and passive is because it's going from high concentration to low. High to low. That's pretty much the key of passive transport, going from high to low concentration. It's a natural. You're in a crowded room. You just want to get out. Doesn't take any effort. Trying to get students into a classroom or a crowded space, you have to push them in there, make them sit there, make them pay high prices, and say, your life depends on this. No. It's a classroom setting. It takes energy. It takes money. Okay, I don't know. Bad analogy, maybe. So just kind of study those pictures. Nothing much to it besides what I've already said. Facilitated diffusion of polar and ionic molecules using a carrier. So we'll describe some of these in a little more detail later. Or you can read and take notes about it. But basically, their conformation or shape changes according to the charges. So here's a lot of writing on a slide. I don't tend to read it all to you. But now we're switching gears to active transport. So things that do not naturally pass through need a little help. Sometimes they're too large or they have a charge because they're polar, and so we have proteins. But sometimes, even so, we need things to go from a low concentration to a high. That's in reverse. So we're going to look at that now. So a pump in the plasma membrane binds and transports a solute against its concentration gradient using energy in the form of ATP, adenosine triphosphate. So it's called the sodium potassium ATPase, depending on where it's talking about, such as in the mitochondrion, or generally the sodium potassium pump, as we'll most likely refer to it. Most vital for maintenance of these concentrations. When you are hot, you feel yourself, you kind of pull your sleeves up and put your arms kind of like this, maybe about a centimeter to an inch. Just leave them there for a little while. Even right now, instantly I can feel a little bit of warmth on each surface of my arm, just being so close. The human is always radiating heat. You're in a crowded place and you touch me like, oh, you feel so warm now. Sometimes you get icy cold. Your blood vessels have constricted. But generally speaking, even then, you're generating heat constantly. How much heat? Well, stick a thermometer in your mouth and you're about 98.6. Now, doesn't mean you are. At any given time, you might be 98.1 or 99.1, 96. So you have within homeostasis, a range of temperatures. But generally speaking, your human body burns enough energy. There's enough chemical reactions to maintain that 98.6 degrees Fahrenheit. And that is primarily due to this protein right here. This membrane protein is in every single cell of your body. Some cells have more than others, particularly the nervous system, for sure, constantly working. And then some muscle cells, of course, have a little more than usual. And then every cell has an amount, because it is the number one way in which to regulate, and it is very sensitive in your kidneys. Oh my gosh, can't wait till we get there. Very sensitive. I mean, just the slightest millivolts of difference, and this pump is setting it off. In this ratio right down here that you see, 3 sodium kicked out for every 2 potassium brought in. Very delicate balance. They're both positively charged, but they don't have the same charging of themselves. So we'll look at that in the next few slides. So we keep these 2 ions against their concentration gradients. We don't want it to be neutral. It's like a battery. A neutral battery is a dead battery. The battery might be happy, but we're not. And you're not alive. So life depends on keeping a difference in charges in order to keep electricity flowing. Yes, you're electric. You see Matrix? All the babies plugged in? That's energy, human energy. So it takes the hydrolyzation of ATP, that is the breaking off of one phosphate group from ATP to make ADP, and we can send that pump into action. And this pump working with all this energy, ATP, is what generates enough heat to maintain ATP. When you get cold, your muscles start to shiver. These things are working. And the sum of all those chemical or metabolic reactions are what keep your body temperature where they are. So this is a very important pump. We got, where are we at? 17 minutes. All right. So you can go through this picture and just study how it goes, but you can see the shape here already. We have a high concentration of sodium and a low concentration of sodium, yet where are we pumping it? Out. We want all that sodium out of the cell. We want it to be right there. We see the higher potassium inside, and yet we're still bringing in more potassium. We're trying to keep that membrane potential, as we'll talk about it later. So to keep it regular requires energy. This protein, membrane protein, only moves, only pumps when ATP is bound to it. Unless there's no ATP, then there's no pumping. So another form of active transport is major. It's not the molecular level or the protein level, but it's vesicles. So vesicles are these small sacs. They're almost the size of organelles inside your cells, and they enclose large portions, and they will move. So because they're being moved by something, transport, chaperonin proteins in the cell, that requires energy. Any movement requires energy in that sense. So small sacs filled with large molecules, too big to be transported by any other means. So it's enclosed in a phospholipid bilayer, and it allows them to fuse or be formed. When we say them to fuse, we're talking about the vesicles to fuse with the membrane and empty the components on the other side. We'll see a nice picture of this here. So here is a vesicle that's risen to the surface of the cell and then fused with it, and it just kind of throws up everything on the outside. So this is the product being released by exocytosis. But let's go back one slide. So exocytosis is how large molecules exit the cell, and that's known as secretion. So when we say pancreas secretes, or your skin cells secrete sweat or secrete hormones or oils, this is what we mean. A cell made a product. That product was tagged to be exported out of the cell, exiting the cell, so exo, outside. So the vesicle fuses with the membrane and spits it out. The vesicles release it. Like I said, there's a picture. So endocytosis is simply the reverse of that. Cell eating. So if exocytosis is putting it out of the cell, endocytosis is bringing it into the cell. So process where cell ingests large particles, just imagine the reverse. Now this is a cool picture because this is a specific kind of bulk transport. You'll see that there's this receptor and marker in bacterium. So one way we eat things, and macrophages will clean up the environment, is when that bacteria binds to a receptor. We say, whoa, you can't be out there, and just engulfs it, fuses with the lysosome, and that lysosome's hydrolytic enzymes will break it down. Hydrolytic bursting. All right, last slide for this. There's another form of endocytosis, is pinocytosis. So phagocytosis was cell eating. Phago means to eat. Cyto, cell. And that's a condition of eating something. Pinocytosis is more of a survival thing. So you're under stress and you just need nutrients, or you need something, anything. Sometimes it'll just engulf a whole big droplet of stuff and hope there's some good things in there. Another one is, again, right here. So we have clathrin-coated pits as an example, but a coated pit of proteins that recognize some certain molecule in the extracellular fluid, and once those bind to it, that's kind of the cell signal. Take it up, and so it invaginates, pinches off, and you form a vesicle that can be called an endosome. And again, it might be hydrolyzed, or it might go on to do other things. If it's not needed, it's just exported. That's transcystosis. So it's endocytized, and then it's exocytized. So rather than say that, we just say it's transcytosis. Or let me move this out of the way. You can get this PowerPoint. All right, well, that's kind of the skinny on transport across the membrane. The next lecture will be on solute concentration, osmotic, and tonicity, which is a little more of the application. But I wanted you to get the basics of cell transport in 22 minutes. So one question to think about, maybe for discussion on a flip grid, is why or how does gargling salt water help a sore throat? Why do people gargle salt water when their throat is sore? Why does it work? Maybe you can relate diffusion or osmosis to that. And we'll talk about osmosis later. Okay. Bye now.